there we are inside your gonads, inside your testes, and you're going to be making some sperm now. What independent assortment of chromosomes says is this, that when a homologous pair of chromosomes, like this pair of chromosome number 1s, is going to be separating into either this sperm cell or this sperm cell, it's random whether maternal goes up to that one or whether maternal goes down to that one. And what each pair does is independent of what any other pairs do. It comes down to a random coin flip. And so I got my coin here, and I'll say heads means the maternal goes up, and tails means the maternal goes down. So heads, and so for your pair of chromosome number 1s, it ends up like that. But the independent assortment means it's independent of what anything else happens. So the other ones don't care what your first chromosome number 1s did. So for chromosome number 2, completely random, happens to be the same way. That goes there, and that one goes there. It's independent, so what your pair of chromosome number 3s do doesn't care what the other ones did. And in this case, it happens to be like this. Paternal goes up, maternal goes down. And for your chromosome number 4 pair, same thing. They don't care what any other pairs do. For them, it's a random thing. Half chance this one's going to go up and half chance it's going to go down. And so there's 2 sperm that you made. And notice they have a mixture of your maternal and paternal genes. And we happen to get, and if you look at the top sperm, maternal chromosome number 1, maternal chromosome number 2, paternal chromosome number 3, maternal chromosome number 4, and the opposite one at the bottom. But you could have gotten something completely different. Let me go click back here. When you're making another sperm cell, well, it's starting off the same. Go there and go there. There we go. So now we're getting something different. That. So notice that these 2 sperm cells are not only different from each other, they're different from that other sperm cell those other 2 sperm cells that we made the first time through. So here's your math quiz for those of you who are mathematically minded. If we just limit ourselves to 4 chromosomes here, how many possible sperm cells can you make? I don't think it's 64. I think that's a little too much. Well, how many possibilities are there for the first chromosome? If you just focus on chromosome number 1, there's 2, right? Chromosome number 1, you could have maternal up, or could you have maternal down? So there's 2 possibilities for chromosome number 1. You could get the maternal or the paternal in any given sperm cell. How many possibilities for chromosome number 2? 2. And chromosome number 3? 2. And chromosome number 4? 16. So if a creature just has 4 pairs of chromosomes, by independent assortment, you could make 16 different types of sperm cells, or 2 to the 4th like that. How many pairs of chromosomes do we have? Not 26, 23. And so for human beings, since each of your 23 chromosomes can assort independently, either maternal up or maternal down, there are 2 to the 23rd possibilities, which I think comes out to around 8 million. So just by independent assortment of chromosomes, males can make 8 million different types of sperm, and females can make 8 million different types of eggs. Now, if you really want to blow your mind, that 8 million is just the number that you make. Your spouse can make 8 million types of their gamete, whether it's sperm or egg. And so how many possibilities are there together? Well, you multiply the 2 8 millions together, your 8 million varieties plus your spouse's 8 million varieties. And I'm not sure what 8 million times 8 million is, but it's a big number. So, you could have trillions and trillions of kids and never have them be identical to each other because of all the variety in your sperm, the variety in your eggs by this independent assortment. kind of mind-boggling. Sound alrighty? Okay, so that's independent assortment of chromosomes. Each pair of chromosomes segregates independently. Let's go back to here for a second. All right. So let me give another example here. That goes like that down like that. I'm not going to bother flipping the coin for a second. But I'm just going to get that, and that, and that, and that, and that. So notice that by independent assortment, you do get your sperm or your eggs having a mixture of maternal and paternal genes, which is what we want because that allows you to have the great variety in your sperm or your eggs, so your offspring will have a lot of variety in them. But notice it only allows you to make mixtures of maternal and paternal genes in your sperm as long as those genes are on separate chromosomes. So I'm saying that if there's a maternal gene here, you can have a paternal gene there. If you have a paternal gene there, you can have a maternal gene there. As long as the genes are riding on separate chromosomes, then you can get it with mixtures of them. But for genes that happen to be located on the same chromosome, independent assortment of chromosomes not mixing them. I'm saying if this maternal chromosome number 1 went right here, all the hundreds of genes that happen to be on that chromosome are all going to be all maternal genes. So it's the independent assortment of chromosomes is good at mixing maternal and paternal genes, but it's not perfect because it can't mix maternal and paternal genes if those genes happen to be on the same chromosome. And so there's this second method that your body has of mixing your maternal and paternal genes that can even mix them even if they're on the same chromosome, and that's this thing called crossing over. And so the purpose of crossing over is to allow a mixture of maternal and paternal genes even if they're on the same chromosome. You say, well, how is that possible? Because the chromosome's either going to go up or it's going to go down. How can I mix those up? Well, let's focus in on your pair of chromosome number 1s here. But what I'm going to show you for the chromosome number 1s doing crossing over will apply to all your pairs of chromosomes. Okay, so when this is a diploid cell because it has 2 chromosome number 1s, and it's about to go through meiosis 1. Before it goes through meiosis 1, it's actually in prophase that this occurs. This is what the pairs of chromosomes do. Each homologous pair of chromosomes stack up on top of each other like this. They actually physically come together inside the cell. And then through a random process, parts of each homologous chromosome can potentially break off and exchange with the other homologous chromosome. And again, it's a coin flip. Will this piece right here exchange with this piece? Well, it might or might not. In this case, it will. So that part of those 2 parts of the chromosomes do crossing over, or synapsis as it's sometimes called. Will this piece exchange with this piece? It won't. Will this piece exchange with this piece? It won't. Will this piece exchange with this piece? Right? It will. So those guys move apart from each other. Will this piece exchange with this piece? I don't know. That's a big guess. There we go. And will this piece exchange with this piece? No. Will this piece exchange with that piece? Yes. There we go. And so notice that this is giving you now, you're creating chromosomes that do have a mixture of maternal and paternal genes. And they'll eventually move apart from each other after they've done the crossing over. So that's not just for chromosome number 1. All 23 pairs of your chromosomes do that. And so in your gonads, in your reproductive organs, you get this crossing over process. And so it's no longer even accurate to speak of your maternal chromosome number 3 and your paternal chromosome number 3 because they've now kind of mixed their DNA up with each other. But this does mean that even for genes that happen to be on the same chromosome, you can get them separated in meiosis 1. And there's all that variety I was talking about. So let's do one more mathematical exercise here. If we have 25,000 genes and each gene can assort independently of all the other ones, that means in any given sperm cell you might have your maternal or your paternal copy of that gene. You have 2 to the 25,000 possible sperm you could make, because each sperm could get a maternal copy of a gene or a paternal copy of a gene. And that's, I don't know what that number is, but it's gigantic. And then your spouse is doing the same thing with her eggs. And so she has 2 to the 25,000 different eggs she can make. And so we multiply these two by each other, and that's the total number of different types of genetic offspring you can make, which I don't know what that is. It's like a universal gigantic number, but it's big. And so that's the point here, that there's a huge variety of offspring even just two people can make with each other. And that's Mother Nature trying to ensure that the offspring always end up very, very different from each other. All right. Now, I found from giving this lecture before that people kind of get it, but the first time through, you may not fully get it. So don't worry if you're not sure. That's normal. One of the purposes of us doing this lab today on chromosomes is it reinforces a lot of these concepts. Oh, maybe I didn't do this part right here. Well, it just sort of goes with that. I think you already got that part there. Okay. Anyway, so you're going to be doing some working with chromosome models today. You're going to get each group will get one of these pieces of white cardboard and this chromosome kit right here. The chromosome kit will include a piece of yarn